So I have the pleasure of introducing Marc Reitman for today's Endocrine Grand Rounds. Marc is a New Englander by heritage. He received his undergraduate degree at the MIT with a double major in life sciences and chemistry before he headed to WashU in St. Louis for his MD and PhD, the latter with Dr. Stuart Kornfeld. He then completed his internal medicine residency at Columbia Presbyterian Hospital before heading to the National Institutes of Health for his fellowship, where he's remained essentially ever since, although there was a 9-year stint where he worked for Merck as their director of obesity studies or something like that. Marc, maybe you can correct me. But he's internationally recognized for his research, which focuses on obesity and metabolic rate and body temperature. He's published over 120 peer-reviewed articles. He's a smart and a good guy and a good speaker. He'll speak to us today about understanding regulation of metabolic rate and body temperature to understand obesity. So Marc, thanks for joining us. Podium's yours.
Hey, David, thank you very much for the invitation and for the very kind introduction. And with that, I'll start. I have no disclosures. All of the work that I'll talk about is public. So we're all interested in obesity, and obesity is a modern epidemic. This shows that there's an inflection point, say in the late 1970s, where the obesity rate went from, say, 15% to 33%. And so we're really interested in this. Why is this happening? How can we fix it? And the reason we're interested in it is not how we look in our bathing suits on the beach in the summer, but it's because obesity is a major risk factor for multiple comorbidities, metabolic disease, diabetes, weight-bearing osteoarthritis, some cancers. And this just is an old slide that just shows that as of 1994, the obesity rate was below 20% in every state of the United States. And by 2013, the obesity rate was over 20% in every state of the United States. And this just shows you the concomitant increase in type 2 diabetes, just to illustrate the fact that obesity is, the prime focus is not obesity per se, but the problems that obesity causes. And as Dr. Harlan mentioned, I spent 9 years at Merck, the first 6 of which I was helping lead the search for a pharmacotherapy for obesity. And obesity pharmacotherapy is really different and really difficult because just like hypertension or dyslipidemia or diabetes, long-term therapy, lifelong therapy presumably is going to be required. So this, but unlike some of those other diseases, the immediate consequences of obesity are kind of mild. So any therapy you invoke has to be incredibly safe. But again, the goal is not just a reduction in body weight, but a reduction in the comorbidities. And there are a number of drugs that are still approved for short-term use, that are not approved for long-term use, that predate the requirement for efficacy studies, and those are listed. But thinking about the long-term treatment for obesity, there's been a number of missteps, if you will. One of the earlier and more famous one is dinitrophenol, which is a chemical uncoupler. Its use peaked from the early to mid 1930s to the point where 100,000 people in the United States were on this drug. And it had a very low therapeutic index. Some of them died, and it was one of the reasons for the passage of the 1938 Food and Drug Act. There are 4 other therapies also listed here, all of which have been approved and then taken off the market for various comorbidity or adverse event reasons. So currently there are 4 FDA-approved drugs with varying efficacy. I think the efficacy is likely to be greatest for liraglutide. However, that's I'd say long-term efficacy is still an open question. We just don't have sufficient experience with it. But the other point I want to make on this slide is that really only 2 of these mechanisms were done, if you will, logically, science-based. Only rimonabant, a cannabinoid 1 inverse agonist, the opposite of marijuana, and orlistat, a non-absorbed lipase inhibitor. These are the only 2 of these mechanisms that were developed rationally, meaning we thought about the science, we said, aha, let's make a drug, and it should have this effect, and it might work for obesity. All the rest of these were fortuitous. These were all discovered because they had effects on body weight in other circumstances or were fortuitously noted to have that and were then developed as obesity treatments. So this really is sort of a quest. How do you rationally develop drugs for obesity? How do you do that? And so my introduction to obesity is that it really is a balance between energy intake and energy expenditure, either body processes or heat generation or physical activity. And what are the causes for the obesity on that framework? And here's a slide I like to sort of show just to make the point that we really don't know what we know. So this is the sales of bottled water in the United States. And you see that it tracks remarkably well with the onset of the obesity epidemic. And so what I want to say is obesity is very easy to study. It's very easy to draw correlates, but it's very hard to know which of those are causal associations. And this is a slide that's one of my favorites, used to be updated yearly by Morgan Downey. He's no longer operates his website, but each one of these links is a putative cause of obesity with a hot link to the PubMed paper that supports this statement. And so what I want to say is that, and these aren't really causes, right? These are associations. But the point is we know remarkably little about what really causes obesity. We have a bazillion associations, we have a bazillion hypotheses, and remarkably little solid data. And that's sort of the driving question that I have and has been motivating my career recently. So what is the situation? The situation is obesity is a mismatch of the intake and energy expenditure. And I would argue, rather than focusing on intake or expenditure per se, we need to be focusing on coupling. This is about the regulated homeostasis. So for example, in the long term, if you go on a diet, your body will lower its metabolic rate to avoid losing weight because that's a threat to an organism, both individually and as a species. Similar, if you increase your energy expenditure by saying going on that marathon, you eat more because your body is sensing that you need the energy stores. So I think the critical questions in obesity are, when is this compensation complete and when is it not? And this means obesity is a brain disease. The control of energy intake and expenditure and its coupling is integrated by the brain. And really what I've done here is frame this in terms of homeostasis, where all this is designed to keep the body functioning normally. But, and again, in the shorthand, there's also the hedonic regulation, where you eat something because it appeals to you, it tastes good. There's something about it that's not necessarily related to its energy metabolism reasons. And so I would argue we need to understand both the homeostatic and hedonic regulation, and we need to understand the physiology and what causes it to fail. So my 9 years at Merck led me to believe that we don't know enough about human physiology to be really approaching obesity in a rational way. And so I've focused on the mouse as a model system. And if you look at, say, intermediary metabolism, the citric acid cycle, it looks like this in the mouse, and it also looks like this in the human. So in that sense, a mouse is an amazingly good model for the human. But if you look at a mouse, there's some ways it may not be. And so this is a mouse, and this is the same scale, human, and you see a mouse is much, much smaller, about 1/3000th the weight. And that leads to a lot of the that is the underlying basis. This scaling is the underlying basis for a lot of the physiology differences that you have to understand in a mouse in order to make a mouse a better model for as an obesity drug discovery and physiology discovery tool. And so very briefly, there's a whole science of scaling, Olametry, and basically it's based mathematically on an efficient network of transport, meaning a capillary, an alveolus, a cell. These are all the same size in a mouse and an elephant. And yet the number of branches of the trachea to get to the alveoli, the number of branches from the aorta to get to the capillary, those are all numbers that do increase with body size. And so there's a whole rigorous mathematical theory that develops scaling factors for each of these various readouts. And the underlying mathematical principle is that you have an efficient branched network to supply nutrients or air and to remove waste products. And just to put a couple of the scaling factors down, these are theoretical numbers, but body mass, a human is 3,000 times heavier than a mouse. But yet, even though you're 3,000 times different in body weight, the core body temperature is similar. Similarly, the blood pressure is similar. On the other hand, heart rate is maybe 6+ fold different. A resting mouse heart rate is 400 to 500. And another thing that's different, and the underlying drivers are that the metabolic rate of the mouse is, for its weight, much higher actually than a human. And it derives, one of the reasons is that a mouse has a much higher surface area to volume ratio. So these body size considerations all underlie thermobiology. So again, to summarize, humans are big, mice are small, yet they have the same temperature. They have the same diurnal rhythm, where you have a 1 degree warmer temperature in the active phase, which is dark in mouse and light in human. A human body core is well insulated and doesn't change much. The mouse body core is poorly insulated. In a human, most of the heat is a byproduct of metabolic reactions, whereas in a mouse, some of the heat comes from metabolism, but you need more just to keep the body warm. So in mice and small mammals, brown adipose tissue does that. Humans have brown adipose tissue, but it's a really minor contributor to metabolic rate. So the physiology of large mammals is devoted towards heat dissipation. Lots of species dependence, all use vasodilation and environmental adjustments, whereas a physiology in small mammals is oriented toward heat generation, such as by brown fat, and then heat conservation by posture or nesting or other behavioral things. So in a large mammal, core body temperature, which is abbreviated TB, is highly and tightly regulated and constant, whereas in a small mammal, body temperature is also just as highly and tightly regulated, but this regulation can allow it or drive it to vary to adapt to situations. And I mentioned brown fat again. There's two basic types of adipose tissue: white fat, which function is to store triglyceride, has all these triglyceride-filled droplets. It has very few mitochondria. When it's activated by the sympathetic nervous system, it releases fatty acids for fuel for other places. In contrast, brown fat is supposed to make heat, has lots of mitochondria, giving it the pink stain in H&E. This tissue is also controlled by the sympathetic nervous system, but what it does is when it's activated, it will expend energy by burning fatty acids and glucose, both from its internal stores and also those released by the rest of the body. And I'm ignoring or lumping the other kind of adipose tissue, beige or bright, which is an inducible brown adipose tissue. And it really, functionally, it's the same as brown adipose tissue. So when I say brown fat, I'm also referring to functional beige and bright tissue also. And humans do have brown adipose tissue, and it can be activated. This is data from Aaron Cypis, just showing that if you give a beta-3 agonist, mirabegron used for overactive bladder, that you can get an increased glucose uptake in the brown fat depots in the neck and along the spine, which is the same as the activation of the brown fat you get by cold. So humans have brown fat, and we can talk later if people are interested in the questions. But I think brown fat is usually a minor contributor to human metabolic rate. So one of the things we were trying to do is sort of bring the understanding of mouse thermal physiology a little bit more into the modern era. And so here's an example of a mouse where we put the mouse at a range of ambient temperatures from 22 up to 33 and back down to 4 degrees, while simultaneously continuously recording their metabolic rate, their respiratory quotient, their physical activity, their core body temperature by telemetry, and their food intake. And by taking all this information and then integrating it, I'll show you some of the ways we've analyzed it. The first is that body temperature regulation in the mouse really has 3 main contributors. The first is, as I mentioned, the light-dark phase, which is about a degree difference. The second is that body temperature increases with physical activity. And third is there is a slight decrease in body temperature with going from a hot to a cold environment. So we've sort of developed a mathematical model for this to do that. But as you can see, the other thing we did was used a method of graphing the data attributed to Scholander, which actually I think was really used earlier by Kleiber. And so we've used their method of analysis in our data, but it's just an incredibly powerful method of analysis. So I thought I'd review it for you. And so what we've done is taken the data I showed you a couple of slides back, done a bunch of calculations with it, and now graphed the energy expenditure versus the ambient temperature. And by energy expenditure, we've removed the effect of physical activity and the thermic effect of food here. We'll add those back later. And when you look at this kind of graph, you see there's a basal metabolic rate, the lowest metabolic rate you observe. And then that metabolic rate exists in the thermoneutral zone. And at the lower end of the thermoneutral zone, the area where the mouse has used all of its passive energy warming capabilities and now has to turn on active mechanisms such like shivering or brown fat thermogenesis. And that transition point is called the lower critical temperature. Below the lower critical temperature, the mouse increases its energy expenditure linearly because it doesn't want to waste energy to get warmer. And when you extrapolate that increase in energy expenditure to adjust to an environmental temperature, you can find the defended body temperature. And what's amazing about this is by just putting a mouse in a different external body temperature, external environmental temperature, you can determine what its defended body temperature is without having measured its body temperature. Now we actually did and showed they were the same, but it just illustrates the power of the technique. And the slope is the heat conductance, which is also mathematically the inverse of the insulation of the animal. This is an incredibly powerful way of looking at the data. And what we've done here is put the real data and use those sorts of analyses to divide it into different components. So if you look in gray, this is the basal metabolic rate, just what it takes to exist. On top of that, you have physical activity, thermogenesis in green, the thermic effect of food, the energy needed to assimilate molecules of food into their place in the body. And then finally, you have this huge amount of blue, which is the cold-induced thermogenesis, the amount of energy expended just to maintain body temperature. And what's remarkable about this is energy expenditure, but in steady state, this is the exact same thing as energy intake. So you look and say, my God, a mouse at 20 degrees or 22 degrees where we're studying them is expending maybe a third of its energy just to stay warm. And when you put a mouse in the cold, it's more like 2/3. So this is really different than humans. And that leads to some different biology. So what we have here, these red lines are individual tracings of core body temperature with an intraperitoneal telemeter sensor. And this is 12 hours of light phase, 12 hours of dark phase. What we did is we took away the food. So you see we take away the food, the mice run around because they're excited. And then they go back to their light phase body temperature. Dark phase comes around, they get warm, but they realize, they really don't have food available. And since we do this on the weekend when nobody's changing cages or making noise. And since they're in a cold environment, and since they don't have a room temperature being a cold environment, they have inadequate food. They go into a state called torpor, where their core body temperature can drop to within about 1 degree or 2 of ambient. And this is normal physiology for all mice, but it's not that frequently observed because people don't look for it. But it can save a huge amount of energy, and it's a rational adaptation to an insufficient fuel source. This is not something humans would do. But okay, so we want to minimize these effects on physiology of a cold environment. So, and we look, and humans really behave as if they have very little cold-induced thermogenesis. So what would happen if you studied mice at 30 degrees? And mice at 30 degrees have roughly no cold-induced thermogenesis, matching humans, and they have a similar distribution of physical activity thermogenesis, thermic effect of food, and basal metabolic rate thermogenesis. So what we've done by taking the mouse where we usually study them at 22 degrees and reduce the cold-induced thermogenesis to roughly 0. And I used to call that thermoneutrality. You see why I don't call it that later in the talk. And the question is, would studying mice at thermoneutrality or 30 degrees be more predictive of the effect of human obesity drug efficacy? And just to show you one example of the studies that we did, this is dinitrophenol again, which is a chemical uncoupler. So what it does is it takes the transmitochondrial proton gradient, which is normally coupled to ATP synthesis, and instead just lets that proton gradient, electrochemical gradient, be dissipated by the dinitrophenol. So now all the mitochondria turn up in order to maintain their proton gradient. And so we use this as a test, and here's the data. So the prediction is, if you study these mice at about 22 degrees, that normally you have a resting metabolic rate and a cold-induced thermogenesis. And now if you add a low dose of dinitrophenol, you'll have a dinitrophenol-induced metabolic rate, and you'll just downregulate the cold-induced thermogenesis. And this is actually actual data. That is what happens. If you look and study the mice at 22, their brown adipose tissue gets turned down, but there's no change in total energy expenditure, no change in food intake, no change in body weight or adiposity or glucose tolerance. Now what we'd say is, what would happen at 30 degrees? Now you wouldn't have cold-induced thermogenesis to turn off. This is actual data. This is the increase in metabolic rate you see. And it turned out there was just a little residual brown adipose activity that was turned off. And interesting, and total energy expenditure increases, but interestingly, there was no change in food intake. I had predicted that food intake would increase to match the increased energy expenditure. And I think this is one example of where we don't understand the coupling between energy intake and energy expenditure. Since the energy expenditure is greater than the food intake, there was a decrease in body weight and an improved glucose tolerance, as you'd expect from a change in body weight. So we'd been playing around with the idea of a thermoneutral zone, an idea, but we started thinking about it and realized we had troubles finding evidence of what happens at the upper end of the thermoneutral zone in mice. And this is a paper from a group, Hill is the first author, where they just looked at energy consumption records for cities as a function of the outside temperature. And you can see here, and again, this is a very intuitive way of looking at it, because you see below a certain, in this thermoneutral zone, you're going to do passive things like wear lighter clothes or heavier clothes, or use low energy things like open windows. Below a certain degrees, 10 degrees here for Ames, Iowa, 10 degrees centigrade for Ames, Iowa, you turn on your heat, but you don't turn on it to make the house 80 degrees or 90 degrees, you turn on to make it comfortable. And so you end up with this linear increase in energy expenditure. And at the warm end, the same thing happens. Maybe you start turning on fans here, very energy efficient, but then you turn on the air conditioning and you see that you have an increased energy expenditure at the upper end. And so we wondered what happens in mice. And this is just 6 different research groups that have published this sort of graph, Scholander plots. And everybody shows a nice thermoneutral zone, then the energy expenditure goes up in every case. But we were looking for primary data, had troubles finding it, so finally we decided to develop some. So we couldn't find any prior studies, and we also were interested in what happens to body temperature when you do this, which had never been done. And we had a big surprise. Our surprise was that mice, again, and this is now dark phase data I'm showing you first, that as the ambient temperature gets warmer, that the mice stop expending any energy. But when you get to what I'm going to call a thermoneutral point, and here D for dark, that what happens is when you get to the point where energy expenditure is minimum, by then body temperature already is starting to increase, and body temperature here is going up about 0.8 degrees per degree in ambient. So this is the point at which the mouse loses control of its thermal profile. The mouse can't control it anymore. This is really bad for the mouse. So what we want to do is define what I'm calling a new concept, the thermoneutral point, which is discrete ambient temperature below which energy expenditure increases and above which core body temperature increases. And the way these are generated, by the way, is by segmented line regression. So this is all based on thousands of data points where this point is assigned by regression, this point is assigned by regression, these slopes are assigned by regression. We have error bars for all of this, and that's all in the original paper. But I'm just drawing the computer fit lines for all of this. So the first point I want to make is in the dark or active phase, mice have a thermoneutral point in the dark, which is coincident with their thermoneutral zone, which is a point. Now, if you look at the light phase, it's a little bit different. What happens is, in the light phase, the mice are maintaining a degree lower body temperature. They hit what I call their thermoneutral point light, at which point they allow body temperature to increase, which until they hit the same regulated dark phase body temperature, in which case they again lose control. So they have a flat energy expenditure, but I argue this is not a thermoneutral zone because core body temperature is increasing. And again, all these points and lines were determined by segmented line regression. And again, and what's kind of interesting is that these lines are completely coincident. The thermoneutral point dark is where body temperature increases in the dark and the light, and similarly the thermoneutral point dark is where energy expenditure increases in the dark and the light. And this is due to the mice running around. This is probably mostly physical activity, probably also some Arrhenius effect, a Q10 effect. So it turns out by looking at body temperature and by looking at this carefully, we've got a much more complicated situation than those simple graphs would indicate. And it has some definite implications. First of all, people have studied the human thermoneutral. Oh, I didn't put the reference on. Sorry, all the references for this, for the human data are in this reference. Apologies. It's old data, often from the 30s. Anyhow, so humans don't have a thermoneutral point. They have a 10-degree thermoneutral zone. And there's no inactive human data that we could find in the literature. So the point is, mice have a thermoneutral point. Humans have a thermoneutral zone. There's fundamentally different thermal biology, which is not fixable, if you will. So I would argue that studying mice at thermoneutrality, quote, is not feasible. Having a thermoneutral point light of 29 and a dark of 33 is not the same as a thermoneutral zone of 4 degrees. And in order to maintain at the TNP light and dark, you would have to change the ambient temperature by 4 degrees every 12 hours, which, you know, with the changes happening quickly and coupled to the light phases. And so this is just not feasible. So there is no way to study mice at thermoneutrality. And so then if you can't study mice at thermoneutrality, how can you make the mice more like humans without feasibly doing it? And so we propose, and again, this is more speculative, is that an ambient temperature of 28 to 29 degrees, so just below the TMP light, is what we would argue for. This allows the mouse to use its thermoregulation, native thermoregulation, because unlike humans who typically live near thermoneutrality, mice live below thermoneutrality. So you want to allow them to regulate. But you still want to stay near the thermoneutral point because it minimizes the cold-induced thermogenesis and tones down at least a little bit the role of the brown fat. Okay, but people have been using 30 degrees to study mice and calling it thermoneutrality. What does this mean? Well, again, to review, 30 degrees is above thermoneutrality in the light phase and below thermoneutrality in the dark phase. And the problem is increasing body temperature, such as you're going to be doing during the light phase, increases the inflammatory and immune actions. So the biggest correlate, if you will, of body temperature, for example, you therapeutic hypothermia, the only problem you get with the hypothermia is that you get more infections because you've toned down the immune system. Similarly, when when we get our second COVID vaccine and get a fever, we're just showing that the immune system is designed to increase body temperature to ramp itself up and therefore ramp up the mechanism of the body. So the conclusion I like to make is, as much as I used to believe this, mice are not just small humans when it comes to thermobiology, and we can't magically make them the same. We can minimize some of these effects, but we can't completely fix them. So let me summarize this portion of my talk and see what my time is. This portion of my talk is that the thermophysiology of human mice is different. Small mammals need to generate and conserve heat, whereas large mammals need to dissipate heat. Humans typically live and are studied under thermoneutral conditions. Mice are typically studied at room temperature, well below thermoneutrality. I showed you a little bit of this. Drugs can have different effects when studied at thermoneutrality versus below it. The mouse thermoneutral zone is only a thermoneutral point, and we propose revisiting the definition of thermoneutrality to include body temperature. The thermoneutral point changes diurnally by 4 degrees, at least under our conditions, and these results suggest major updates into how mice are studied and used to model human physiology. Now, I have another section that gets into the physiology. And when I was at Merck, so David, I have 40 minutes or an hour?
Oh, let's say 45 minutes.
Okay.
Because we like to leave time for questions.
Sure. So what I'll do is I'll give you a little bit of insights. So this is getting at the mechanisms that we can study this. So when I was at Merck, one of the mechanisms we studied was something called BRS3. BRS3 turns out to be an orphan G protein coupled receptor. And what happened was bombesin is a frog peptide. It has nothing to do with mammalian biology. And we still don't know what the native ligand for this GPCR is. But it's expressed in the brain, and I'll tell you a little bit about that. Because when the knockout mouse was made, the knockout mouse both became obese by a reduction in metabolic rate and body temperature and an increase in food intake. And it actually also showed an increase in body temperature and blood pressure, making one hope that if you made a drug to this, that you would have a really beneficial effect on multiple axes. And so at Merck, we developed a very pretty agonist, which was single-digit nanomolar across species, and that allowed us to study the biology. And just to show you that this drug in mice increased body temperature in the wild-type mouse, but not a knockout. It decreased food intake in the mild-type mouse, but not a knockout. And it increased metabolic rate in a wild-type mouse, but not a knockout. And it also caused weight loss in mice. And what's interesting about this is that certain weight loss mechanisms will cause you to get dangerously low in weight. So if you give a cannabinoid inverse agonist, you have to stop the experiment because the mice lose too much weight. What's interesting about BRS3 is the mice plateau at a safe body weight. So you can't, presumably you can't overdose it on body weight at least. So the effect of this BRS3 agonist really was the opposite of the effect of the knockout mouse. However, when it was taken into humans, it actually increased blood pressure unexpectedly. So that caused Merck to lose a lot of interest in this, and I had left for the NIH a while, prior to we published this, and therefore I ended up deciding to study what the mechanism is to try and understand this. And so the question is, where and how are BRS3 agonists acting? And I'll skip this. But basically to say that if we delete BRS3 in glutamatergic neurons, we reproduce the full global null phenotype for body weight, food intake, metabolic rate, temperature, insulin resistance, and response to a BRS3 agonist. Similarly, if we took the full knockout and re-expressed BRS3 only in glutamatergic neurons, we got a full wild-type metabolic phenotype. So this was our proof that it was really working in the brain that hadn't formally been shown before, and suggested that glutamatergic neurons were major importance. We then made a mouse, a BRS3 Cre mouse, that allowed us to study BRS3 Cre in certain areas of the brain that we were interested in. But what's pretty about this is that it's not all over the brain. This gives you some hope that you're going to get some specificity. And so what I'm going to show you first are some studies in the DMH and the PVH, the dorsomedial hypothalamus and the paraventricular hypothalamus. And basically, I'm going to show you the different functions of these two areas. And we used chemogenetics or DREADS to selectively activate or inhibit just those specific neurons we wanted to attack. And this just shows you how that was done. What we did is we injected a virus into the DMH that allows you to express a GPCR that only responds to CNO, a drug that nothing else in the body responds to. So we achieve a neuron chemotype by the BRS3 expression. We achieve anatomic localization by where we put the virus. And we achieve activation of timing by giving an agonist injection. So this just shows you we can inject and express this GPCR only in the DMH. And when we activate it now, and again, so this is 12 hours of light phase, showing you if we give a CNO injection, we give vehicle in black or CNO in red, that we get an increase in body temperature, an increase in metabolic rate with no change in physical activity. So this says that activating the BRS3 neurons in the DMH regulate energy expenditure and body temperature and not physical activity. If we did the same thing in the BRS3 neurons in the PVH, we now see no effect on body temperature, no effect on energy expenditure, no effect on physical activity, but now we can suppress food intake in data I'm not going to show you. And we go back to the DMH and now give an inhibitory stimulus. Here we're now coupling the CNO to an inhibitory DREAD. You see now we have bidirectional control. So, and I won't show you, but this also controls that also controlled blood pressure and heart rate. So we've identified two populations of BRS3 neurons with non-overlapping functions. The PVH BRS3 neurons suppress food intake with no effect on body temperature, energy expenditure, physical activity. The DMH BRS3 neurons increase body temperature, heart rate, energy expenditure, and activate brown adipose tissue via the sympathetic nervous system with no effect on food intake or physical activity. And the DMH is usually thought of as an effector pathway that talks to something called the raphe pallidus, which then talks to the intermediate lateral spinal cord neurons, which are the sympathetic preganglionic neurons. So we established that the DMH neurons are talking sympathetically to the brown fat, and that the DMH BRC neurons are also programmed by the preoptic area, which is a central region of the brain. That's the computer for regulating body temperature. And so more recently, stuff I'm really proud of, we've now gone upstream a little bit to the preoptic area and asked what in the preoptic area is a BRS3 neurons doing? And the answer turns out to be a lot. So first of all the preoptic neuron types identified to date will reduce body temperature upon activation. And in general, the preoptic is a very inhibitory neuron concentrated region of the brain. So what we showed was that if you put the mice in the cold and look for which neurons are activated in the cold, about half of the preoptic BRS3 neurons are activated by the cold. So then we studied the function of these neurons the same way, and we find that if you activate the preoptic area of BRS3 neurons, they increase body temperature. This is the first neuron type to do that, and it's bidirectional. If we inhibit them, they decrease body temperature. And the other thing these neurons do is they turn out to, the point I want to make is that this is a single injection of drug that lasts a long time. We now also did the same thing using something called optogenetics, where instead of using an injection of a drug to activate these neurons, we use a optical fiber and a laser to do it. And the beauty of this is that, you know, these mice are hooked up to the fiber the day before, they're not being disturbed. So now you can study things like heart rate and blood pressure without handling the mouse, which is an incredible confound. And you see, again, if you look and turn on the laser for 20 minutes, you get the same increase in body temperature from the preoptic area. But now, instead of doing this for 20 minutes, we do this for, say, 20 seconds. And look at this. You see that with very short pulses, you know, the body temperature hasn't moved, right? But even with pulses as short as half a second or 1 second, we can start seeing increases in heart rate and blood pressure. So these neurons are directly talking to the sympathetic output to the heart. And we did pharmacology showing that it was sympathetic and not parasympathetic. And the last surprise we got from these neurons was the following. What we did was we used a technique called tetanus toxin, which permanently inactivates neurons without killing them, without causing major trauma to the brain. And so what we do is we give tetanus toxin, and now what happens, the diurnal change in body temperature overshoots cold in the inactive phase and overshoots warm in the active phase, again, with no change in physical activity. And this happens whether it's 8 days after you've induced the tetanus toxin or 5 months after. You can see the standard deviation of the daily range in body temperature is massive. And so what we think these neurons are doing is acting, well, I think they're having a governor function, that they're removing the fine-tuned control of body temperature. Another possibility is that these are two separate classes, one that governs high temperature range of neurons and one that governs low temperature range. And then what we did is we mined data from Dulac and collaborators of single-cell RNA-seq and said, what kind of BRS3 neurons are there in the preoptic area? And it turns out if you do this clustering analysis, you find that there probably are at least six types of neurons in the preoptic area. 1 excitatory neuron type and 5 inhibitory neuron types. And our next goal is to see if we can now subdivide the BRS3 neurons and attribute these individual functions, individual classes of neurons. So to summarize, I made this as a build. All right. So to summarize, we've shown that there are BRS3 neurons in the preoptic area that control heart rate, body temperature with no effect on physical activity. I didn't show you this, but the other populations work in the opposite direction. There are lots of collaterals of these neurons, unlike the more downstream pathways. But at least one of the pathways is that the preoptic area to the DMH talks to both body temperature and heart rate. So these are cold defense neurons. These are neurons that are orchestrating the body's response to a cold environment. Since the mice have a normal body temperature, they're not the only way it happens, but that's, at least with acute stimulation, it's clearly one of their functions. The second function they have is they modulate body temperature variability with overshoot and undershoot if they're inactivated. And there are multiple classes of BRS3 neurons, maybe 6. And with that, I'd like to summarize and say obesity is a brain disease with dysregulation of energy intake expenditure coupling. Mice are generally great models for understanding diabetes and obesity. Compared to humans, mice have different thermal biology. They have a significant cold-induced thermogenesis. They use torpor. They have a thermoneutral pointer, some of the examples I showed. Mice near thermoneutrality may be a better model for testing efficacy of drugs for obesity. I didn't show you the data for this, but CNS active anti-obesity drugs in humans are always advertised as anorectic agents. But in mice, at least, they work to reduce food intake and increase resting metabolic rate in mice. So I think that all the CNS active drugs everybody talks about and uses are actually affecting both sides of it. The thing about the humans is humans' food intake is a much more important factor than metabolic rate is, and the mice are more evenly divided between those mechanisms. I showed you that BRC neurons in the PVH regulate food intake, BRC neurons in the DMH regulate energy expenditure and body temperature, and they're part of the effector pathway from the preoptic area. And BRC neurons in the preoptic area regulate body temperature, including defense from cold and fine-tuning body temperature regulation. They're probably more of the higher order function neurons is our hypothesis. And I'm really bad at acknowledging people, so I want to say that all the neuroscience was driven by Ramon Pignol, an amazing postdoc in the lab. He had help from postbac student Colleen Hadley, Alison Mogul, Haley Provence. The thermoneutral point data, this is the work of Wojciech Skop. I have a couple long-term collaborators. Everything in the lab I do in collaboration with Oksana Gavrilova and her mouse metabolism core facility. Chuying Zhao is a staff scientist in my lab who made a lot of the mice we're using. And Michael Crashes teaches me neuroscience every time I talk to him. And Kevin Hall and his postdoc, June, were instrumental in the hardcore statistical analysis that we derived for the thermoneutral point analysis. And with that, I'd like to open it for questions.
Thanks, Marc. Terrific.